Hello, everyone. My name is Raymond Suhandynata. I'm a recent ComACC Clinical Chemistry Fellow graduate, and I am still at UC San Diego Health, and I'm here to tell you a little bit about the work I did during my fellowship, which is the title of this talk is COVID-19 Antibody Testing in the Pandemic, Lessons Learned. So, as many of you know now, SARS-CoV-2 is a virus, and here we have an illustration of the viral architecture of this virus. As you can see, when a virus is attempting to infect your cells, there is an immune response mediated by B cells, and antibodies are generated. As you can see, there are two major antigens on the outside of the SARS-CoV-2 virus, the spike protein, also known as the S protein, as well as the nucleoprotein, also known as the N protein. And these are the two major viral antigens that serology assays, clinical serology assays have been developed against. So back in early 2020, we at UCSD Health were asked, can we validate a SARS-CoV-2 serology assay? Mainly because, as we all know, PCR-based assays are great for acute infection, but what about past exposure to the virus? So we were first tasked to determine if they were indeed reliable and could be used in the clinical setting. So we in fact did this. We partnered with a local company, and we were fortunate enough to get one of the first analyzers to perform a SARS-CoV-2 IgG antibody assay. This was an antibody assay directed against the spike and nucleocapsid protein. And as many of you know, working in the clinical laboratories, we are very focused on the robustness of each assay. Here we have a table, which the header is the between run CV, the within run CV, and the total CV for the IgG assay. On the left column are the samples that we tested, the positive QC and negative QC that came with this kit, as well as different patient pools. The patient pool high, patient pool low are from SARS-CoV-2 PCR positive patients, whereas the patient pool negative are from, these were stored plasma or serum samples from patients from 2018, frozen at minus 20 degrees Celsius. So we consider these SARS-CoV-2 naive patients. And as you can see, we saw very good precision between run, within run, and total for all of these samples or patient pools. The only one where we had a large variation in the CV was this patient pool negative. This is at the time was because we were essentially measuring noise, and as you know, when you're measuring noise, then the precision suffers greatly. So while this isn't exactly what we wanted to see, it was expected since this assay essentially would go down to zero, would report out zeros. We also, at the time, were very concerned about cross-reactivity to other types of respiratory viruses, as well as non-COVID coronavirus. So here we have a subset of different samples from different patients and infected with different respiratory pathogens, as well as different immunological diseases, HIV, ANA, anti-DS, essentially lupus here. So as you can see, we didn't get any cross-reactivity, which was very important and gave us some confidence moving forward with this assay. So we also then decided to take a look to see if we could detect seroconversion in SARS-CoV-2 confirmed patients. These are PCR positive patients, and we had longitudinal samples and serially monitored them through the course of disease. So in the left, we actually also have, I didn't show you the precision data for it, but we also had an IgM assay which looked at IgM antibodies against the SARS-CoV-2 and spike protein. And it had similar precision as the IgG assay. As you can see, when you look at these patients over time, there's a point when through the course of their disease that they seroconvert. And the point where this assay deems that to be a patient is positive is 1, and this is a semi-log scale. And as you can see, the median time to IgM seroconversion is 5 days, with an interquartile range of 2.75 to 9. Interestingly, we saw that the IgG seroconversion was in fact 4 days, which is at the time was very confusing. We are educated that IgM comes before IgG during the humoral response. It was a little bit confusing at the time. However, many other studies have reported a similar effect, that they see IgM and IgG almost come concurrently, which is a little different from other serology, the serology of different diseases. So essentially, we're seeing a seroconversion on this assay of about 4 days. One thing that we were heavily questioned on is what about the prevalence? And as we all know, prevalence does affect the positive predictive value and negative predictive value of all clinical tests. So we did some math, essentially, based off of the sensitivity and specificity we were observing in our cohort. And this, based on the sensitivity and specificity of each assay, in this case, the IgM assay for SARS-CoV-2, we calculated the positive predictive value and negative predictive value at different prevalence levels. And as you can see, for IgM, IgG, as well as IgM plus IgG, around a 1% prevalence rate, we actually have really poor positive predictive value. And we really focus on the single-digit prevalence levos because there were some discussions on whether we should be screening the general population or whether or not we should only be using it for patients presenting with symptoms. And at the time at UCSD, we had a estimated prevalence or PCR positivity rate of about 4.3%, as in 4.3% of the patients being tested were actually positive by an RT-PCR test. However, if we were going to go to the general population, we assume that to be much lower. And we could already see, if we're looking at a 1% level, our positive predictive values are quite poor, less than 70%. And in fact, for IgG in this panel is less than 50, about 50%. So one of the things that we try to educate on is how positive predictive value works. So as you can see here, the SARS-CoV-2 antibody test, even assuming a great test, 100% sensitivity, 99% specificity, in a test population of 1 million with a prevalence level of 5%, disease prevalence of 5%, there would only be 50,000 disease individuals in this population. But based on a specificity of 99%, you'd have a 10,000 individuals that would just be false positives, just based on the math alone. So putting in together, and this is the formula that is used to calculate positive predictive value, which is the number of true positives divided by the number of true positives plus false positives. We would get a positive predictive value of 83%, roughly. And that's not bad, but that's at a prevalence level of 5%. However, what about if we took a different scenario and we looked at the same population with a prevalence level of 0.5%? In this population, we have only 5,000 disease individuals, but the same number of false positives. And what this does is it reduces our positive predictive value to 33%, which is quite awful. So it is just basic math, but it is helpful to visualize it, especially in the context of screening low prevalence populations. So we were very proud to contribute in this field. As you can see here, I'm showing you the website for the Journal of Applied Laboratory Medicine, the highly cited collection, where we have one of the most cited papers, or the most cited paper. I'm not sure. I don't think there's actually a ranking, but still, it is nice to have our work recognized. And if you're interested, here's the citation in the DOI. So moving from this study, we were interested in how to improve the positive predictive value moving forward, especially as we begin to test low prevalence populations. So, well, we were fortunate enough to be to have sort of first dibs, one of the first dibs on the Roche Cobas and Abbott Architects SARS-CoV-2 serology assays. So for those of you that don't know, the Roche Cobas is an automated chemistry analyzer where at UCSD Health, this is essentially our workhorse where we do most of our patient testing. We also at UCSD Health have an Abbott Architect where we do a lot of immunosuppressants and NT-proBNP, et cetera, that we had access to as well. And both of these vendors had SARS-CoV-2 specific antibody tests. So in this study, we looked at the individual performance and how each of these assays would, the positive predictive values that you would expect to see from each of these assays in a clinical setting. And again, much like we did previously, we saw really poor predictive values in using either assay, any of these assays, either Diazyme, Roche, or Abbott. Some are ranging from 30 to 60%, depending on the prevalence, depending on the assay, and at a 1% prevalence level. So this got us thinking, how can we make this better? So what we decided to do was look at the discordant specimens across each platform. And these are discordant samples. In this case, there's patients, these are PCR positive patients, positive for SARS-CoV-2, labeled 3 through, well, ranging from 3 to 57. However, right below it, there's 4 other patients. These are patients who had their plasma or serum drawn in 2018, and these would be COVID naive. And these were patients where we saw discordance across each of these platforms. And while it is interesting to look at the differences in the positive patients, the most important thing for when you're trying to screen a low prevalence population are your false positives. So we looked at the false positives, and we saw that this particular patient negative 78 from 2018, had a false positive on the Roche assay, which was not a false positive on any of these other assays. So we decided to look at the effect of doing a screen and confirm approach, which would mean that basically using orthogonal assays, or in this case, different assays, to help confirm initially screen positive SARS-CoV-2 serology results. So in this case, if we're using a screen and confirm approach, we actually see really great positive predictive value when you use the Roche total IG assay and confirm with either the Diazyme or Abbott assay. As you can see here, in this case, because in our particular cohort we had an NPA or specificity of 100%, we had no false positives with the Roche and the Diazyme combined, or the Roche and the Abbott combined. We actually get 100% positive predictive value. And this is because the single false positive we observe on the Roche would, in this particular cohort, would have been ruled out as a false positive, and therefore we would not have reported that out as a positive result. However, the Diazyme and Abbott IgG assay, they share a lot of, well, more false positives, and they're not exclusive. They share false positives. And what happens there is you don't see the increase in positive predictive value that you see when you're combining the Roche with the Diazyme or the Roche with the Abbott. And that is important. So it is important to understand, are the false positives shared between each assay? And if there are, then even though you're using different assays, it doesn't necessarily mean you're getting the increase in specificity that you're looking to get. So at the time, the CDC was also working on this, as we all know, and they in fact came up with some guidelines. And at the time, they put out this bulletin, and they determined there was no advantage for using IgG, IgM, or IgG, IgM, or a total antibody assay. And they also came with a guideline to not test low prevalence population, and in fact to use an orthogonal testing algorithm when testing populations with low disease prevalence, which is exactly the conclusion that we came to in our study. So with that said, we actually went live back in April, I believe. Or sorry, not April, September. That April was for when we went live with the Diazyme assay all by itself, September. This is when we went live with our screen and confirm approach. So in this case, we would screen for positives on the COBOS, and we would confirm positives on the Diazyme. And in this case, live testing began September 1st of 2020, and we had our reporting stream set out as such, either positive, negative, or indeterminate. And so the workflow was such as if a patient was screened positive on the Roche, they would go to the Diazyme, and if the Diazyme said they were negative, we would report that as indeterminate. And if they screen negative, then these specimens wouldn't get confirmed on the Diazyme. They would just be reported as negative. So our next question was, do antibody results mean anything? And this was a huge question at the time, because what do these serology tests mean? So we partnered with some scientists at the Scripps Research Institute, and who had some experience with neutralizing antibody assays. And so what are neutralizing antibodies? Neutralizing antibodies are antibodies released from your B cells, and their job is to neutralize an antigen. And in this case, the idea would be neutralizing the spike protein, which is known to be the main method for how SARS-CoV-2 infects humans. And in this case, if this spike protein, which is labeled by, in this case would be this red, the little spikes coming out of this red virus, the antibodies would bind the spike protein and prevent viral entry into cells. So our collaborators developed a pseudovirus assay where they had the SARS-CoV-2 spike protein, or S protein, on the outside of a pseudovirus containing luciferase, and had cells expressing the human ACE2 receptor, which is known to be the mode of entry for SARS-CoV-2. And inside those cells, there's more, I think it's luciferin. And what happens is if the virus gets into the cell, light is generated. However, if there are neutralizing antibodies on the that are able to bind the spike protein and prevent it from interacting with the ACE2 receptor, viral entry is not observed and no light is generated. So it's a light-based assay that is measuring the ability of a pseudovirus to either get in or not get in, because we incubated the pseudovirus with patient plasma containing neutralizing antibodies. So here we have our cohort. It's in this case we had a positive cohort containing 31 patients and a negative cohort containing 100 patients. The 31 patients are PCR positive, and they had a total of 87 specimens, whereas the negative cohort had 100 specimens and mostly from individuals from 2018, but some individuals that were positive on a respiratory panel nucleic acid test for other things such as rhinovirus, other types of coronaviruses, etc., or other healthy individuals from 2020 that were not infected with SARS-CoV-2. So in this case, we evaluated this pseudovirus assay from our collaborators. We determined that the assay is quite robust, especially with SARS-CoV-2. They also had a SARS-CoV-1 assay, and these individuals infected with SARS-CoV-2 had some cross-protection to SARS-CoV-1, which is not surprising. But since we were mainly focused on SARS-CoV-2, we moved ahead there. And in this case, we have bar graphs where we have the percentage of samples with neutralization or neutralizing antibodies. And that was characterized as samples with neutralizing titers greater than 50, which essentially means you can dilute the patient's plasma 50-fold and still get 50% viral, prevent 50% of the virus from getting into the cells. So we also, in a similar fashion to our previous studies, we looked at the neutralization titers of PCR confirmed patients, SARS-CoV-2 PCR confirmed patients over time. As you can see, we have 13 different patients here monitored over up to 25, 26 days. And these are their neutralization titers. And they follow a very similar trend to their antibody levels as well. So this helped us gain some confidence that the clinical serology assays at least seem to mirror or correlated, at least in this small cohort, to the neutralizing antibodies that are that we so desperately want to prevent severe disease and infection. So moving forward, we decided to do a retrospective evaluation of seropositive individuals screened at UCSD Health. So this was a huge study where we retrospectively looked at the effectively 9,530 SARS-CoV-2 serology tests, and we focused on the seropositive individuals because we wanted to see, one, are these true positives? We knew that based on the math, we expect about 50% of our IgG seropositive individuals to be false positives. We wanted to see if that was actually true. And so we did this by looking at what these 164 IgG seropositive individuals would, how they would end up, what would their results be on the Roche and Abbott serology platforms. So this really helped us confirm that there was a real benefit to the screen and confirm approach. So as you can see here, we have a graph showing on the y-axis the positive % agreement with SARS-CoV-2 neutralization. And we evaluated this for the 164 initially confirmed seropositive individuals on the Diazyme assay, and following a screen and confirm approach with either the Roche total IG assay or the Abbott IgG assay, we were left with a 96 positive or 93 seropositive individuals. And as you can see, the positive percent agreement with SARS-CoV neutralization went up from 50%, which matches our 1% prevalence level that we assumed at, to nearly 80%. So this really does underline the benefits of a screen and confirm approach. So here we also have, just for for illustration, we have the values observed for samples with and without neutralizing antibodies in all of these samples. And as you can see, in this case, the Diazyme assay really performed well with its specificity, as in if a sample didn't have neutralization activity, the Diazyme assay more or less didn't have that much of a response. However, the Roche and the Abbott, there's a little bit more spread out. The response was a little bit more varied, as in there were samples that were negative for neutralizing antibodies, but had very high levels. And that could be totally correct. Maybe that individual had antibodies against SARS-CoV-2, but they just weren't neutralizing in nature. And same with the Abbott. And we helped visualize this by looking at the median levels of the positive of the patients who had samples that were positive for neutralizing antibodies, and look at the median on each of these assays. And what we see is that if we use the median as a cutoff for each of these specific assays, we actually have really high specificity for the Diazyme assay compared to the Roche and the Abbott. So one of our last questions was, can we distinguish vaccination from natural infection? So what we did was we looked at two of our tried and true assays, the Diazyme IgG and Roche N assay, which at the time we called that the total assay, but that total assay is only against the nucleocapsid protein. But during the start of this study, Diazyme and Roche came out with new assays. Diazyme came out with a functional neutralizing assay called the Diazyme neutralizing antibodies assay, and Roche came out with the Roche S assay, which is a total antibody assay that is directed against the spike protein. So here we have 4 different sets of box plots, and in this case, we looked at patients that were stratified according to when they received a positive SARS-CoV-2 PCR result. As you can see, very, very similar values we see among all the assays and the response. And the longer as disease progresses, these values become higher. So we also did our own individual clinical evaluations of the Diazyme neutralizing antibody assay and the Roche S assay. However, due to time, I won't be showing that data too much. So we did compare these assays, and looking at the positive percent agreement to a individual receiving a vaccine, we see that we have good positive percent agreement with all the assays besides, with both of the new assays, but not the Roche N and the Diazyme IgG. Now, we were really surprised on the Diazyme IgG assay because initially at least from the manufacturer, we were told this was an N and S assay. However, this data really suggests it's more of a nucleocapsid assay versus a spike protein assay as well. The Roche N assay, we don't actually expect it to pick up any of vaccinated individuals because vaccinated individuals are only exposed to the spike protein. That is what either the mRNA vaccines are expressing or the adenovirus-based vaccines are expressing, or even the attenuated viruses. And in this case, the Roche S and the Diazyme neutralizing assays, those do pick up vaccinated individuals, and that makes sense. The Roche S assay is developed against the spike assay, so we expect that to pick it up. And the Diazyme neutralizing assay is a functional neutralizing assay, which is looking at the ability of patient plasma to inhibit the interaction with the spike protein and the human ACE2 receptor. So one may ask, what is this one individual that's positive on the assay? Well, it turns out that individual actually was positive for an active SARS-CoV-2 infection. So this person was also naturally infected, as well as vaccinated. We then looked at, in a similar fashion, individuals that had already received their booster shot. So what I was showing you previously were individuals that had not received their vaccine booster. Now that we look at individuals that had received their vaccine booster, we see that we had a 100% positive agreement with the Roche S assay and 94% agreement with the Diazyme neutralizing assay, which essentially means that these two commercial assays can pick up vaccinated individuals fairly robustly. In this case, we then looked at the neutralizing antibodies with our pseudovirus-based assay that were produced by vaccinated individuals. And as you can see, we have, in this case, a table looking at pre-booster individuals and the neutralizing and the presence of neutralizing antibodies in those patients or in those individuals. And in this case, we had 24 vaccinated individuals, and of those 24, 21 were positive on the neutralizing assay. So we had a PPA of 87%, roughly. And here we decided to look at post-booster. Well, if people wonder, does the booster make a difference? It definitely does. In this case, we're looking at the neutralizing activity of individuals post-booster, and we had 71 that had received their boosters, and 70 were positive on the pseudovirus-based neutralizing assay. So people wonder, should I be taking my booster? Yeah, it does make a difference. We definitely see a response, which I'll be showing you a little bit more, will be illustrated by this next slide. So in this case, we have on the y-axis the log base 10 SARS-CoV-2 ID50 titers, and on the x-axis we have the days post initial injection of receiving their first dose of vaccine. You can see individuals in the pre-booster that have not received their boosters, they are just single dosed. They're sort of all over the map in terms of their neutralization titers. The line where we determined that they have neutralizing activity was 100 in this case. This is a log base 10 scale, so that's 2. And 3 individuals here hadn't formed sufficient neutralizing activity, the ones that had not received their booster. However, those that had received their booster, only 1 individual, and they had just received their booster here at this triangle that is at day 25. They had just received their booster. Everyone else formed very robust neutralizing titers, except this individual. Unfortunately, we weren't able to follow that individual for an extended period of time to make sure that they had formed neutralizing antibodies. But we definitely see a distinct change in the neutralizing antibody titers following the booster shots. So those 3 individuals here did not make neutralizing antibodies, and this single triangle did as well. So we also decided to look at the correlation of commercial antibody assays with pseudovirus-based assays and the neutralizing antibodies that are detected by them. And what we found is we really compared only the Diazyme and the Roche assay, the S antibody assay, the Diazyme neutralizing assay and the Roche antibody assay, because those are the only ones that we had that picked up vaccinated individuals. And we found that the Diazyme neutralizing assay had a really strong correlation, positive correlation with SARS-CoV-2 ID50 titers. The Roche S antibody assay also correlated, however, it was not quite as robust at 0.39. So in this case, we definitely prefer the Diazyme neutralizing assay. So that brings us to our summary and conclusions. So first off, all of these platforms, the Diazyme, Roche, and Abbott assays, can be used to detect past exposure to SARS-CoV-2. It's important to remember that positive predictive values are poor when screening low prevalence populations. That's true of any clinical assay, not just these serology assays. And in order to really be able to robustly perform serology testing in low prevalence populations, a screen and confirm approach should be used. This should help your PPVs be, close to or even above 90%. And the neutralizing assays, antibodies that were detected, were much more approved when you're using a screen and confirm approach. In fact, about 80% of individuals determined to be seropositive after a screen and confirm approach had neutralizing antibodies. And the Roche S and Diazyme assays robustly detect infected and vaccinated individuals, which is important to know because you can actually use the N and the N nucleocapsid specific assays, or in this case probably the Diazyme IgG assay, even though it's supposedly a S assay as well, to differentiate infected versus vaccinated individuals, because vaccinated individuals will only have antibodies against the S protein versus naturally infected individuals will have antibodies against the S and N proteins. And we found that the Diazyme neutralizing antibody assay correlates strongly with PSV pseudovirus ID50 titers in vaccinated individuals, which makes that a good assay to consider if you're trying to get some meaning out of these tests. So again, what does this all mean? There was a relatively recently published article on neutralizing antibody levels, and that they are highly predictive for immune protection from symptomatic SARS-CoV-2 infection. And here I just listed a few of the conclusions from that assay, from that study, which is the antibody half-life is about 60 days, and the neutralization titer is about 108 days, and that significant loss of protection from infection from SARS-CoV-2 happens around 250 days. However, importantly, protection from severe disease is retained, which is likely due to the role that memory B cells and T cells play. So with that, I'd like to acknowledge my colleagues at UCSD Health, specifically Robert Fitzgerald, who is our ComACC Clinical Chemistry Fellowship Director, as well as all of these great individuals, as well as our collaborators at the Scripps Research Institute, as well as the commercial vendors that supported us on this journey. So with that, I'd like to conclude the talk and hope that everyone was able to learn something. Thanks for your attention.